Hello guys, this side Dr. Masman here, and today we will start biochemistry. I will be telling the whole biochemistry in a very concise manner, and I will be talking about first enzymes, then collagens, and then we will go to chapter wise, right? So let's start, and we start with the holoenzyme. So what is holoenzyme? It is the combination of two, apoenzyme plus coenzyme. Apoenzyme means P is coming in between, right? Apo, P. P means protein, right? Then comes coenzyme. What is coenzyme? When apoenzyme was protein, coenzyme is non-protein, right? So non-protein has two factors, cofactor and prosthetic group, right? So let's see the cofactors first. So what are the cofactors? So we have around 8 cofactors. First one is zinc. See, remember in this zinc IN. IN means insulin. So what does it secrete? It secretes insulin, right? And also think of alcohol in that. So we have all the dehydrogenase and peptidases, right? First is alcohol dehydrogenase, then we have lactate dehydrogenase, then we have carbonic dehydrogenase, anhydrase, then carboxypeptidase. So when you have carbonic NIC, okay, also in zinc we have INC. So that's how it can be similar rhyming sounds. And also this question has come many times, carbonic anhydrases, right? Then comes copper. Copper means just think of Y. Whenever you have Y in anything, that leads to copper. So tyrosine, okay, lysyl oxidase, okay, cytochrome oxidase, cytosolic superoxidase dimutase, okay. So these are the 4 things about copper. Then comes 2 diseases, Wilson disease and Menke disease. In Wilson disease, okay, it is autosomal recessive, and also it has ATP7B. In Menke disease, it will be in male because it's Menkes, that is male, okay, and this is X-linked recessive disease, and this is ATP7A, okay. So just think of Menke, it's man, right? So both things are coming down, so copper will be down in it. When it comes to Wilson means W, it is up, so means copper will be increased in Wilson disease, okay. And also just remember one thing, 3-methyl histidine, this will be there in Wilson disease, which will be decreased, right? It's just a question. Then comes molybdenum. What is molybdenum? In this, what we have to think is MO and X, right? So OX will be there in any of the enzymes. Whenever you see OX, that will lead to molybdenum. So for example, XO means xanthine oxidase. So X and O is there, xanthine oxidase, that is molybdenum. Then when it comes SO means sulfite oxidase, that is also molybdenum. So these 2 things are there which comes under molybdenum. Then comes iron, means MSD, Mahendra Singh Dhoni, right? So M, S and D. M means malonate, S means succinate, D means dehydrogenase. These are the 3 things which comes under iron. Then comes nickel. What is nickel? In nickel we will see urease, okay. So nickel means Nic, both Nic will come, but there will be urease in it, so nickel means urease. Then comes calcium. So in calcium what you have to see, calcium, CaLCi UM. So LCI is LCI is there, na? So we need to know about lipase, Li, okay. So that will be lipase, and then lecithinase in calcium, okay. Then comes selenium. What is selenium? Just remember one thing, in selenium we need to know the thio, okay. Like we go to Thailand, okay, THIO, sale. When we go to Thailand, we sell some things, right? Or we buy something. So just we remember we need to sell the thing. So selenium is sale and thio is Thailand, right? So the enzyme, it will be glutathione reductase, okay. Thioredoxin deiodinase. So IO and thio, the both things are there in selenium, right? And whenever selenium deficiency is there, so that will lead to a disease that will be called as Cassan's disease, right? Then comes 2 things, either it will be magnesium or it will be manganese. So whenever it is magnesium means Mg, M is mutase, G6 means G6 phosphatase, G means, right? And whenever it comes manganese means man, so MAN. M is mitochondrial superoxide dimutase, okay. So in copper it was cytosolic superoxide dimutase, but here it will be mitochondrial superoxide dismutase, okay. And then comes A is arginase. Then comes N is ribo N nucleotidase. So MAN, both of them, all 3 are there in manganese, right? Then if manganese and magnesium both are together, then we have, like for example, we have both energies will be there, kinetic plus potential energy. Okay, so Ke, kinetic energy, both will be increased in it. So Ke means K is kinase, E means enolase. Okay, then comes about micro element and macro element. So in micro element, we need to know some, and for macro also we need to know some of the elements, right? So in micro we need to know FeCu, FeCu. Okay, then comes MnMo, Manmohan, then comes Ja, Zn, then comes Be, that is B, then it's nickel, NiCl. So FeCu, Manmohan, Ja, Be, nickel. FeCu means FeCu, Manmohan means MnMo, Ja means Zn, Be means B, nickel means NiCl. So this is what we need to remember about micro elements. And regarding macro, it's कौन किसका मांगे प्यार. कौन means CHON is कौन. किसका means KSCA. CA is the one which has already come in the exam. Then मांगे means MG. Then comes प्यार means phosphorus. So CHON, KSCA, MG and P. So this was about macro elements, right? Okay. And also in macro elements, just remember one thing: O-linked glycoprotein is there in it, right? Then comes collagen. So this was all about the part which I have told you about the cofactors. Now let's proceed to collagen. What is collagen? See, it's a protein. It has a quaternary protein, and the most common in collagen we have is glycine. Okay, then we have 4 types: type 1, type 2, type 3, type 4. In type 1 we have, because it's 1, right? So bone, BON. Okay, so that will be type 1. Type 2 means 2 will be there. So means cartilage. So means just say 2, cartilage, right? That will be cartilage. Then comes vitreous humor. So don't say vitreous humor, just say vitreous humor. Vitreous humor, that will be 2. Then comes 3. So there also 3 will be there in it. Just for example, artery. So don't say artery, just say R3. Okay. Then comes granulation tissue. Okay. Tissue means also 3. It's starting from letter T, that means 3. Then comes fourth. Fourth means floor. So what will be in floor? It will be basement membrane. So this is how all 4 types of collagen are done. If someone asks you what's the most common collagen, then it will be skin. Okay. Then comes 2 things: either it will be extracellular or it will be intracellular. So whenever you have extracellular, so cell. So whenever you have cellular in it, C is there. Okay, in between. So just make it to a bond which is a covalent bond, which is also start from C, covalent cross link. Okay, and whenever you have intracellular, that means I and TRA. TR you have, okay, just take the TR and make it triple helix, which is also called as fibroblast. Okay, then comes myofibrils. Okay, myo, okay, that means 6. Okay, so whenever they ask about myofibril, that will be 6. So about myofibril, we have components and we have some defects. So whenever it's 1, okay, 1 means O is coming, right? O means all the diseases start with the O. Then it will be osteogenesis imperfecta, osteoporosis. Then we have Ehlers-Danlos, which is type 7. Then also 1 is coming. See, O or 1 is coming in 1. Osteoporosis ka O. Okay, osteogenesis imperfecta ka bhi O. Then comes Ehlers-Danlos. In this we have type 7. So 1 ka bhi O. So that's how defect 1 is there. Then comes type 2. In type 2 we have osteoarthritis. Okay, where it will be, see, arthritis usually in the knee. So we have 2 knees, right? So it will be, it will come under 2. Okay, then we have chondrodysplasia. Chondro means, then also it's dro means do. Do means type 2, right? Then comes 3, defect number 3, that will be Ehlers-Danlos type 4. Okay, see, we also had in defect 1 also we have Ehlers-Danlos, but that was type 7, ONE. Okay, but in 3 we will have type 4. Okay, just come once more in Ehlers and then it will be type 4. Okay, then comes in defect number 4 we will have L-Port syndrome. Okay, and then comes defect number 6, that will be myopathy. Okay, so these all are the defects. Then a question comes about collagen 17, collagen number 7. So about 17 means you will have some disease. Okay, just count the disease, letter of the disease, and you will get to know which one is 7, which one is 17. For example, we have epidermolysis bullosa junctional. Okay, so epidermo means E is 5, bullosa means 2, and junctional is 10. So that will all lead to collagen number 17. And whenever you have epidermolysis bullosa dystrophica, D means 4, okay, and then 5 plus 4, so that's how that you will make 5 plus 3, that can be 7. Or what you can do is collagen number 70 just minus 10, and then you will get 7. So at the end, whenever it's dystrophic, it's collagen number 7. Whenever it's junctional, it's collagen number 70. Okay, then comes collagen. In this we have vitamin C. Okay, in C we have OH, that means hydroxylation. So in hydroxylation we will have two things, either it will be proline or lysine. Okay, and in the proline and lysine, and then you see the Cu, that will be lysine oxidase, right? So because it was collagen and vitamin C, so also this will be Cu, so lysyl oxidase. Now there is one clinical symptom which we need to know about. It is in clinical, first there will be decrease of the vitamin C, and whenever vitamin C will decrease, so starting from the letter C only, one thing will decrease, that will be collagen. Collagen will also decrease. Okay, when collagen will decrease, that will lead to blood vessel 3 type, and that will be fragile, means you can break it easily. Okay, so that means there will be gum bleeding in it whenever it will be fragile, and as gum bleeding is there, that will lead to a disease called scurvy. Okay, and the main thing in this is time. Okay, in this the prothrombin time will be normal. Okay, whenever you have collagen. But now let me tell you about another thing. In that the collagen time will be different. Okay, so what happens is vitamin K antagonist, we have dicumarol and warfarin. So these two things are there, they inhibit, and then comes vitamin K. Whenever vitamin K will decrease, that will lead to carboxylation. Okay, that also starts with K, or it's just C, remember it with K. Okay, and then comes carboxylation. In carboxylation, what you have is glutamate. Okay, so in glutamate, what will happen, for we remember, okay, gamma, glutamate, so GGG, all these things are coming. Okay, so that why it will be gamma carboxylation. And then when it comes to glutamate, there is a factor which is 2, 7, 9, 10, or you can remember as 10, 9, 7, 2. So these are the clotting factors of vitamin K. Okay, and also in this, the main thing is whenever there will be bleeding with vitamin K, the disease will come is hemorrhagic disease, which will be there in a newborn. And the most important thing, what I told you in vitamin C, the prothrombin time was normal, right? But here the prothrombin time will be increased. Okay, in vitamin K. So this is one of the differential diagnosis. In vitamin C, prothrombin time is normal. In vitamin K, it is increased, right? Now let's come to liver cirrhosis, which is type 1 or type 3 defect, that will be collagen. Okay, so you have GLUT, right? Then comes GLUT. What is GLUT? Glucotransporter protein channel. Okay, in this we need to know some of the specific steps. 1 and 3 we know it will be brain, RBC and placenta. Okay, but whenever it comes to type 2, GLUT2, then type 2, what you will have is BKL. Okay, K is what? K is kidney. Okay, L is liver. But do not confuse with B, because B usually student confuse with the brain, but it is not brain, it's beta cell pancreas. Okay, in GLUT2 you have beta cell pancreas. Okay, then comes type 4. In type 4, what you have? You have insulin dependent, okay, adipocytes. Okay, in adipocyte, then what you have in type 4, you have heart and skeletal muscle. Okay, this is what in there in type 4. Then comes 5. Okay, in 5 you have is 5, I means intestine. So in 5 you will have intestine. Then just know about the physiology. What you already know is, because in this there will be facilitated diffusion in GLUT type. Okay, and whenever it's SGLT, then it's sodium glucose link transport, that will be secondary active transport. Okay, this I think you know already. Then comes to amino acid. Let's know something about amino acid. In this, first of all, we have 22 amino acids, 20 plus 2. Okay, so let's start from the basics. So what all we have here is, we have acid. So we have two acids, glutamic acid and aspartic acid. Okay, then these two acids are there. Then comes the amide. Whenever amide comes, these changes into glutamic acid. Acid is there, so this will change to aspargine. Okay, and whenever it's aspartic acid, that will change to glutamine. Okay, one is aspargine and glutamine. Okay, and in this a question can come, which one is the most acidic? So the most acidic will be aspartic acid, right? And also I saw one of the question like, which of the following prevents the aggregation? that will also be asparagine or glutamine, right? Then comes second is sulfur. In sulfur you have two things, MC. Okay, M is what? M is methionine, C is cysteine, and also you can see all these things in the pulses, and also there is a mnemonic PCM. P is pulses, C is cysteine, M is methionine. It's there in sulfur. Then comes hydroxyl. What is there in hydroxyl? See, we know there is one mnemonic and it is TSH. Okay, and TSH means T is threonine, S is serine, H is hydroxyl. Okay, these three things are there for the hydroxyl. Then comes basic. Okay, in basic what you have here is this is most important. In this question always comes. Okay, in basic we have HAL, HAL. H is histidine, A is arginine, L is lysine. Histidine, arginine, lysine. Because see, what I told you in the acid was the most acidic. So that was aspartic acid. But here question can come, which one is the most basic? Most basic will be arginine. Okay, which is HAL is histidine, arginine, lysine. Okay. Then comes, they can also ask like about serotonin, which can be 5-hydroxyl tryptamine. A question has already come. Okay, then fifth one is aliphatic. What is aliphatic? In this, we need to know about GAVLI. Aliphatic means Ali Gavli. Ali Gavli. Okay, Ali Gavli. Gavli. Let's start. What is this? G is glycine, A is alanine, V is valine, L is leucine, iso is isoleucine. Okay, so these are the 5 things which I told: glycine, alanine, valine, leucine, isoleucine. Okay, so in this they can ask you which one of the following is the branch chain amino acid. So whenever they say branch amino, it will be only VLI. Okay, V is valine, L is leucine, and I is isoleucine. Okay, then comes amino acid. What is there in amino acid? In amino acid, see, amino acid. Okay, so what we know is here is they are arophytic. That means pha means arophytic. That means pha means phenylalanine. Okay, tri means tryptophan, and then in tri just put T and Y together. That means tyrosine. Okay, so these 3 things will be there in the amino acid: phenylalanine, tryptophan, tyrosine. Okay, this is very important. And also they can ask you which one of the following absorbs the light. That will be tryptophan. Okay, and these 3 are together combined, and they are a part of benzyl ring, right? So this was about aromatic. Then comes amino. Okay, amino is proline. In immunoproline we have 221 and 22nd. This is the important one which we need to know. Okay, because see, in the 21st generation, what we know the Selena Gomez, right? So we have here selenocysteine. Okay, and in the 22nd what we have is pyro means pyrolysine, because in 21st generation we used to see Selena Gomez, and 22nd generation we are there, so we are the fire, right? So we are firing, so that means pyro, that is pyrolysine. Then comes essential amino acid. In essential amino acid we have a question definitely comes about which one of the following is essential, which is semi-essential, and which is non-essential. In the essential we have try this VIP mol. Okay, I will tell you about one by one all the names, and then I will tell you which one of the specific which has already come. Try means tryptophan. Okay, try this. In this we have T is threonine. Okay, H means histidine. Try this means HIS is there, that is histidine. VIP means V is valine, I is isoleucine, P is phenylalanine. Okay, then comes try this VIP, then comes mol. M is methionine. Okay, A is arginine, L is leucine, and L is lysine. Question has come, which one of the following is all except comes. So there was a question in the essential amino acid. So all the options were there, which I have already told. But there was a question about aspartate. So the aspartate will not be option in essential amino acid because it is a non-essential amino acid. So from this only essential amino acid, you need to take out, first of all, I will tell you about semi-essential, but I am telling you there will be confusion between 2 of them, tryptophan and threonine. Okay, so tri is tryptophan and TH is threonine. Okay, now take 2 of them from the essential amino acid and let it come in semi-essential. So in this try this HIS is there, take out histidine. Okay, and in that you have mol. A is arginine, so take out the arginine, take out histidine and arginine, both of them, and they both are called as semi-essential amino acid. Okay, histidine, this has the maximum buffering capacity. Okay, so both of them are very important. Then comes the non-essential one. See, in non-essential you have two things: either it will be ketogenic or it will be glucogenic. See, just remember, in glucogenic, all remaining whatever you have will be there in glucogenic. Just remember about ketogenic. In ketogenic, one thing is cat. Keto means cat. Okay, cat means leucine. You usually in the ancient era we used to call Lucy to the cat, right? So here it will be leucine. This question has also already come in our exam. Ketogenic means Lucy, that is leucine. Okay, so which and also then it comes in both of them we have TIP. T is tryptophan, threonine, tyrosine, okay, isoleucine and phenylalanine. So these are the ones which are the common. Okay, then comes amino acid. So right now, like I have told you everything in short detail about what all things are there. So now we will be studying hopefully about more amino acid, right? So now comes amino acid, right? In amino acid, what do we have? See, we have arginine. In arginine, what we have is arginine means 9. Okay, that is converted into nitric oxide. Okay, nitric oxide will be what? This is the nitric oxide means NO, and arginine also 9. So N to N, this both are combining, and the enzyme in which both of it will be converting will be NADPH2. Arginine 9 to NO, nitric oxide, NADPH2, right? Then comes tyrosine. Okay, sine means IN, that will convert it to tyrosinase, and that will lead to melanin. So this question has also already come. Tyrosine, when it will convert, what it will form? So INE, tyrosine, and melanin, NIN. So it will convert to tyrosine, and the disease will be melanin. Then comes tyrosine hydroxylate, okay, catecholamine and thyroxine. So INE is coming in all. Catecholamine, INE, thyroxine, INE. Okay, in catecholamines you have three things: epinephrine, non-epinephrine, and dopamine. This question has also come. Okay, then comes histidine. What is histidine? It is decarboxylation and histamine. See, histidine, DINE, take out DINE and let it make it decarboxylation. Then comes histidine, HIS, make it histamine. Okay, which one hypersensitivity it is? It is type 1 hypersensitivity. Okay, and also what it has mast cells. Okay, so about histidine I have told you about arginine, tyrosine, I have told you histidine. Then fourth one is tryptophan. Okay, tryptophan. Tri means 3. 3 means vitamin 3. Then comes to. So whenever it comes to tryptophan, to means it will make it two things: melatonin and serotonin. So whenever tonin will come, okay. So melatonin, serotonin, this will be also secreted from the pineal gland. Okay. And also I told you about serotonin. This will be 5-hydroxytryptamine. So this question has already come, which I already told you. Now let's see about cycles, right? So we have two cycles: cytosol plus mitochondria. Okay. In cytosol, what do we have? We have porphyrin. We have PUBG. Okay. In cytosol, we have PUBG. Okay. And that PUBG means P is porphyrin, which is heme. U is urea cycle. B is beta oxidation of fatty acid, that will be mitochondria. Then comes G. G is gluconeogenesis. Just remember this neogenesis because you may confuse with other words also. Okay. Then comes cycle. So in cycle, what we have is we have mitochondria. Okay. In mitochondria we have inner mitochondrial membrane. Okay. In inner mitochondrial membrane we have cardiolipin, phospholipid, and also in inner mitochondrial membrane we have electron transport chain. Okay. Then that will lead to mitochondria matrix. Okay. And when you have matrix, TRIX, that will lead to TRI means tricarboxylic acid cycle, which is TCA, that will lead to Krebs cycle. Okay. And keto is meto. So whenever you have ketogenesis, that means all things will be in the mitochondria, and all remaining cycles will be in the cytosol. So this is what I told. Okay. Now comes ETC. In ETC we have NADH.H plus. Then we have FADH2. Then we have SLP. Okay. Whenever we have NADPH plus H, means see H plus S. Okay, means there is something which is also adding. So means it is 2.5 ATP. and whenever we have FADH H2, only H2, nothing is adding. Okay, we have only when you have H2, then you have 1.5 ATP. When something is adding, then it's 2.5. When it's not adding, it's 1.5. Then comes SLP, substrate level phosphorylation. Okay, in substrate level phosphorylation, there will be something. First of all, this will give some P, and then that will lead to ADP to ATP, right? Then comes complexes. So this is also important complexes which we need to like what inhibit what. Okay, so we have like 1 2 3 4 5. We have 5 complexes. Okay, so let's talk about first complex. First is CONE1. Okay, so first take the O, that will be oxidoreductase, N will be NADH dehydrogenase. Okay, so these are the complexes, and that this will lead to inhibit what I told about 1. So phenobarbital, ONE, that is 1, right? Then rotenone, ONE, okay, piperacillin. So this was all about 1. Then comes the second. So SECOND. So take S and take the D of the end. S is succinate, D is dehydrogenase. So this was the complex. In this, what will be the inhibitor? This will be malonate and malonate coenzyme A inhibitors, which we also told as MSD. So we have malonate, we have succinate, and we have dehydrogenase, right? Then comes third. Okay, in third we have cytochrome reductase, RD. Okay, so whenever you have RD means R means reductase. Okay, and this will lead to C1, BC1. Okay, and this was the complex. And whenever you want to see the inhibitor, then it will be antimycin, phenolphormin, and BAL. So in this, what we need to remember is 3 things. That will be BAP, right? So in third, what I told about, RD means reductase. Which reductase? Cytochrome reductase. Okay, and also in this there will be C1 because it will inhibit C1. So that will be C means cytochrome, RD means reductase, cytochrome reductase. Then we have BAP. Okay, BAP means B is what? B is BAL, BAL. A is N3-mycin, antimycin, but we will call it N3 because it's 3. And then P is phenolphormin. Okay, then comes 4. 4 means 1 plus 3 equal to 4, right? So there will also be 4 inhibitors here, and also it will be oxidase. See, third was cytochrome. See, cytochrome will be common in third and fourth, but because it was RD, so it will be reductase, and here it will be OFOUR, so O means oxidase. Okay, so in which there will be 1 plus 3 means A1 minus A3. Okay, and there will be 4 inhibitors, CO, CN, azide, and H2S. Okay, these 4 things will be there. Then we have fifth particle. In fifth complex we have is FO and F1 particle. Okay, in this there will be ADP will be converted to ATP. Okay, with there is something in between is SLP. Okay, and in this will be FO and F1 particle is there. So FO means O means oligomycin. Okay, so this will be FO, and then comes etracystyloside. So A will be there, so that means ADP. Okay, so this was all about complex inhibitors. So now let's talk about ET, electron transport coupled oxidative phosphorylation. Okay, so means couple means husband and wife should have been there. Okay, then what we know here is H2O means O2 plus delivery of electron. Okay, at the end is couple means you need to remember is ATP will be produced. Okay, whenever it's couple, ATP will be produced, but whenever it is uncoupler, so ATP will be decreased. There it was produced in couple, here ATP will not be produced. So question comes about the examples of the uncoupler in which ATP is reduced. So we have 3 things here. In 3 things we have, first is chemical uncoupler, second is pharmacological uncoupler, third is physiological uncoupler. So whenever we have chemical, so just remember CAP, mnemonic, CAP. Chemical also start with C. Okay, CAP also start with C. Okay, in CAP what we need to know is dinitrocresol. C is there, so cresol, right? Then comes A is aspirin. Aspirin means high doses. Okay, then comes P is phenol. Phenol means also dinitro. C in chemical coupler, first one was what I told was dinitrocresol. Okay, in P also there will be dinitro, in C also there will be dinitro, but in C there will be cresol, in P it will be phenol, and A is the aspirin which we give in high dose, right? Then comes pharmacological. Okay, whenever it's come pharmacological, then we always say vali. Okay, so that means valinomycin and G gramicidin. Okay, so and this one about among all of them, the one which is the most important is the physiological uncoupler, because a question has already come from it. And first one is thermogenin, which is brown fat from the newborn. Okay, this is the question which already come. Which of the following is the physiological uncoupler? And they will give you some options. Then is thyroxine. Okay, then comes free fatty acid, which is long chain. Okay, and fourth will be bilirubin. So I am repeating again: physiological thermogenin, thyroxine, free fatty acid, which is long chain, and fourth is bilirubin. So this was all about coupler and uncouplers. Okay, now let's talk about the enzymes. Okay, in enzymes we have kinetics, holoenzyme, apoenzyme, and coenzyme. Okay, in holoenzymes we have, I'm just telling you the question which has already come. Okay, in this there will be metalloenzyme in holoenzyme, and whenever it's protein it will be metal activated enzyme. Okay, so if they ask metalloenzyme, that is holo. If they ask about protein, that is metal activated enzyme. Okay, it has the same thing. See, substrate is there, it will form a product. In between there will be an enzyme. Okay, so let's talk about the factors. If there is concentration of enzyme, that means that will be proportional to velocity. Whenever there is substrate, that is also proportional to velocity. But there is something which is inversely proportional to velocity, that will be product, right? This question has also come. Which one of the following is the inverse proportional? That will be product. Okay. Now we know about this. We have Vmax, which have we have velocity. Okay, in the y-axis. In the x-axis we have concentration. Okay, in concentration we have Km. Km is Michaelis Menten constant. This question has also come. Okay, and Km value will be Vmax by 2. This question has also come. Okay, and whenever they ask about what is Km, Km is 1 by affinity. Okay, so whenever they can also ask you about there, so there are two things: temperature and pH. Okay, temperature will be 40 to 45 degree temperature, which is optimum, and the pH, which is optimum, is 7. So in this there will be a curve, okay, of Vmax, okay, regarding like temperature or pH. So the curve name is, which shape of curve will be there, so that is bell shaped curve. Okay, now come the allosteric side. So what, yeah, also one more thing about this which I wanted to tell you is, in the Vmax and substrate concentration, what the curve which we get is hyperbolic rather than parabolic. Okay, now comes the allosteric regulation, which is S shaped allosteric. So S, ALLOSTERIC. So we have S, take out S, this will be shape curve. Okay, which will be sigmoid curve. Okay, so bell shape was the normal one. Now in allosteric there will be sigmoid curve. Okay, then come that these three things. This question always comes from here, is the enzyme inhibition. Whenever it's competition, see, whenever you have competition, you will run the kilometers, right? So Km will increase, Vmax will be same. Whenever it's non-competition, you don't have any competition, you will not run, Km will be same, but velocity will decrease, because you are not competing and you are just having rest, so your velocity will also decrease. Okay, but when you are uncompetitive, see, in non-competitive at least you are competitive, but right now you are non-competitive, so that's why Km will be same, velocity will be decreasing. But when you are uncompetitive, you are not at all in a race or anything, you are just sleeping and just enjoying, then Km will also decrease, Vmax will also decrease. Okay, then comes two things: either it's competitive, non-competitive, uncompetitive. You can see the graph. Okay, this can also come, and there is also a line which is called as Lineweaver-Burke plot. Okay, so I have told you about the graphs. Also, whenever like it's crossing, it's cross. Okay, competitive. Whenever it's not crossing from one point only, both of them are going, it's non-competitive. Uncompetitivity means it's always only parallel, nothing is there, two of the lines will go parallel, right? Then comes isoenzyme. In isoenzyme we have creatine kinase. We have three things are there in isoenzyme: 1, 2 and 3. Okay, in 1 we have brain. Okay, 2 we have heart, and 3 we have muscles. So see, just remember two things: BB, MM. In first you have BB means brain, in third you have MM means muscles, in second combine both of them, M and B. Okay, So whenever you have already done brain, muscle is also done. So what is remaining? Heart is remaining. Okay, so it will be heart. They can ask you about the marker of the reinfarction. Marker will be troponin. Okay, in heart we have marker which is CK2, that will be troponin. Okay, and if they ask you about best marker, that will be MI. Okay, and then comes the lactate dehydrogenase, which is there in the cytoplasm. This question has also come, where does the lactate dehydrogenase is there, so that will be in the cytoplasm. Okay, now let me tell you about, we have some types of these markers. Okay, then we have type 1, 2, 3, 4, 5. Okay, in type 1 what we have is, we have heart, we have RBC, but if you see the H, because we have in type 1 we have heart, so there will be H will be very more, so that will be H4. In type 2 we have heart brain, in 3 we have brain spleen skeletal muscle, and then come type 5. In type 5 will be skeletal muscle, that will be M4. Okay, muscle is there, so M4, heart is there, so H4. Okay, and then comes it like first is H4, then H3. Hello, regarding MI, so what they can ask you about flipped LDH ratio. So in this MI, I means 1, LDH also 1. So 1 will be there and 1 will be more than 2. Okay, then comes alcoholic liver cirrhosis. Whenever come this, then it will be 5. See, because whenever we drink, we are alcoholic, we drink paua. Okay, paua is 5, that means 5. Okay, so in alcohol liver cirrhosis, 5 will be more than 4. Then comes hemolytic anemia. In hemolytic anemia, there will be 2, which is higher than the normal, right? Then comes central dogma of life. In this we have Francis Crick, which is 1958. This was the model in which DNA converts to RNA, then to protein. Okay, then we have 2 things, RS transcription and translation. So let's understand what is this. Okay, so what is DNA? See, first of all, DNA will convert to RNA. Okay, this is a normal thing, and in this, the one which will be converting DNA to RNA will be transcription. Okay, in the RNA, this we call as HNRNA. HN means heteronuclear RNA. Okay, then comes post transcriptional modification, because transcription is already done. Okay, now post transcriptional modification will come, that will make mRNA, from HNRNA to mRNA, that will be post transcriptional modification. Okay, in mRNA there will be tRNA and ribosome. 2 things will be there in mRNA, that will be tRNA and ribosome. Then mRNA will do translation, and translation will convert it into protein. Okay, let me repeat again, DNA to HNRNA. In this there will be transcription, then there will be post transcriptional modifications that will lead to HNRNA to mRNA, and mRNA there will be tRNA and ribosome and tRNA that will translate to protein. Okay, so after that, because see, after transcription there was post transcription modification, here it will be post translational modification. Okay, in post translational modification what we can see is collagen, we can see hydroxylation, we can see, okay, clotting factors we can see. Okay, these all are the things. Now let's see in the post transcriptional modification, which I told you about HNRNA. Okay, in HNRNA it was mRNA, and then comes about the types. For example, 5 to 3 /. Okay, whenever comes 5 to 3 /, then it will be called as splicing, right? Because 5 / we call it as capping or 7-methyl guanosine. Okay, and 3 / is poly A tail. Okay, so basically what is splicing? Let me tell you, it's divided into 2 things. First of all, there will be removal of intron and ligation of exon. Just remember intron and exon. Okay, and intron means what? Whenever you are inside, then you don't code. Means whenever you are inside, there will be non-coding sequence, and whenever you have exon, means whenever you are outside, you code the things. So whenever you are inside your home, you don't code, you just relax, enjoy, non-coding. Whenever you are outside the home, exon, then it's coding sequence, right? Splicing, it is done by whom? it is done by spliceosome, which is also called as nerves, right? So we are done with this. Now let's see what are small nuclear ribonucleoprotein. Means whenever DNA converts to RNA, it is DNA dependent RNA polymerase. Whenever it is RNA converted to DNA, then that will be RNA dependent DNA polymerase. Okay, and whenever DNA converts to only DNA, then it will be replication of DNA polymerase. Just remember one thing: dependent will come through which you are converting. For example, if I am converting from DNA, then it will be DNA dependent. If I am converting from RNA, then it will be RNA dependent. Simple. Okay. Then comes protein. Whenever protein changes, from here question has come. Whenever protein changes, if there is no defect, then there will be protein foldings. Okay, and those protein foldings are called as chaperones. Okay, protein foldings are called as chaperones, and whenever there is a defect, okay, means protein is degrading. Okay, defect is there, means so that will be broken down by whom? Ubiquitin and proteasome. So a question has already come on. First of all, no defect means protein folding, which is chaperone, and whenever it's broken down means protein degradation, that will be proteasome. Question has come about proteasome also, right? Then comes structural protein. In structure we have primary, then we have secondary, we have tertiary, quaternary. Okay, so let's see the primary one. In primary we have linear. Okay, and also linear, in this there will be it will be linear, and in this there will be another forms also which we can see, and it will be like alpha helix. Okay, and we have then beta pleated. So these two, alpha helix and beta pleated, they both will come in secondary. Okay, primary we have linear, and alpha helix and beta pleated, they both are secondary. Okay, also a question has come, which of the following is peptide form? So we can say primary one. And which of the following not participate in proline? Then also we have this structural proteins, right? Then a question has already come on the tertiary one. Okay, so what is called a tertiary? Tertiary is the one in which there is 3D dimensional. See, tertiary means 3. Okay, 3D dimensional. So we call it as MAL. M-A-L. M means myo, A means albumin. M-myo, A-L means albumin. Okay. Then comes quaternary. From quaternary also question has come. In quaternary there is 2 pp chain, but question was there in which we need to tell like it will be either homo or heterodimer. Whenever it comes about dimer, it will be quaternary. And also we have all the things are there, but in quaternary there will be no peptide bond. Okay, so this was the thing. In that it was proline. Here it will be peptide bond. Okay, now let's talk about vitamins. Vitamin, this is the most important part. First of all, we need to know which of the following are fat soluble, which of the following are water soluble, which are antioxidant, and how it is synthesized. Okay, so in fat soluble we have KEDA, K-E-D-A. Okay, in which K is vitamin K, then E vitamin D, and vitamin A. If they ask you about water soluble, which of the following are soluble in water? So you have K3. Okay, K3 means you have W water solubles. So make W in a different position and you will get a 3, K3. Okay, and the remaining will be water soluble, that is B and C. Okay, and then comes antioxidant. A question has already come about antioxidant, which one of the most potent. So A, C and E we have antioxidant, in which question was about the most potent, which is E. Okay, then how the vitamins are synthesized? K3, B3, D3. Ka but D. K3, B3, D3. Okay, now comes the vitamin K. A question has come in which the answer was like a prolonged use of antibiotics can lead to vitamin K deficiency. So this we need to remember. And in this there will be, I already told something about vitamin. First of all, it will be hemorrhagic disease of newborn. Okay, and in this the platelet will be high. Okay, PT, sorry. Okay, and in this there will be mother milk will be there, which will be decreased. Okay, and also in this there are 3 things: fluoro, K1, K2 and K3. K1 is first, so it will also start with F, that is phylloquinine. Then comes K2, Okay, then comes that is menaquinone. Okay, then comes 3 means menadione. Di means 2, 1 means one means 1, 2 plus 1 is 3. Okay, that is water soluble, synthesized in body. And also K is more than D, so that's why it will be in this. There will be also, they can ask like uh, a mother is there which is vegan, so in that what do we use? We use vitamin 12. Okay, so I have told you about vitamin K, and also factors were 10, 9, 7 and 12. Then comes vitamin E. In vitamin E, what do we have? We have alpha tocopherol. Okay, so what I told about vitamin E, it is a fat soluble. Okay, vitamin K I told, and also about vitamin E, what I told, it is the most potent, right? So it's alpha tocopherol. Okay, and it is an antioxidant, it is anti-aging, it won't lead to age if you are using vitamin E, and most potent, which I told. And about vitamin E, if it decreases, then it may lead to hemolytic anemia. This question has also come, which of the following can lead to hemolytic anemia? That will be vitamin E. Okay, and also there will be progressive peripheral neuropathy, PPN. So in vitamin E, what do we have? Tocopherol, antioxidant, anti-aging, most potent, hemolytic anemia, plus PPN, progressive peripheral neuropathy. Then comes vitamin D. In D also we have 3, D1, D2, D3. D1 is calciferol, D2 is ergocalciferol, D3 is 125 di OH cholecalciferol. See, calciferol is common in all. In D1 only calciferol, in D2 ergocalciferol, in D3 125 dihydroxy. Okay, that is also called as calcitriol. Okay, in vitamin D we have skin, liver and kidney. Okay, now first of all sunlight comes. Okay, then what happens? In skin we have keratinocytes. Okay, and then from skin, in skin also we have an enzyme. This is called as 7-dihydroxy cholesterol. This question has already come. And also they say that whenever this is converted, okay, then this is converted to whom? So 7-dihydroxy cholesterol convert into cholecalciferol. This question has come. So vitamin D forms which to which? Okay, then it will cholecalciferol. This will go to liver, and what it will form? It will form 25 hydroxylase cholecalciferol. So this was the thing which was when it will go to liver, this will form this. When it comes to kidney means 2. So it will form 2 things: 1 alpha hydroxylase, 125 dihydroxy, and cholecalciferol. So these things will be formed in kidney. Then comes the function of vitamin D. So in vitamin D, we have function is calcium, and first we need to know the normal value of calcium, which is 9 to 11. Okay, then comes, see, 3 things are here. So do not confuse in these 3 things: intestine, kidney and bone. Whenever you have intestine, calcium will be just absorbed. You have intestine, calcium will be just absorbed. Okay, but whenever you have kidney, okay, just remember, whenever you have kidney, that is calcium will be reabsorbed. Okay, and whenever you have bone, then it will be formation. So intestine is absorption, kidney is reabsorption, and bone is just formation. Okay, then comes vitamin D. If the vitamin D is decreased, then it will lead to what? First is children, that is adult. In children, that will lead to rickets. In adult, it will be osteomalacia. Okay, in rickets it will be like widening, cupping, spring, all these things. Now I am telling you the main things of multivitamins in the whole biochemistry. Okay, vitamin A is the best source. Okay, and it has halibut fish liver oil. This is also a question. Okay, green leafy vegetable. Okay, then comes 3 things. We have retinol, retinoic acid. These 3 things are there. A question has come that if you have if beta carotene is there with provitamin, then it will lead to what? It will lead to 2-retinal, active vitamin A, 2-retinal, AL. Okay, so just remember this one. And also in this there will be green leafy vegetable. Question has come on this also, which I told already. Then comes functions. Okay, in functions, what do we have? Here we have antioxidant, growth of epithelium, we have reproduction, we have germ cell, sperm. Okay, then we have rhodopsin. Rhodopsin we have, in this we have opsin plus 11 cis retinal. Rhodopsin means D is there, right? So it will be dim. Dim will be light, means rods will be there. Okay, when you have dim lights, then rods are functional. In rhodopsin we have opsin plus 11 cis retinal. When you have trans retinal in this, then it will be sodium chloride, that will lead to hyperpolarization. Okay, in vitamin D what do we have? We have infertility, dry skin, xeroderma, okay, xerophthalmia, okay, we have nyctophobia, fear of dark, keratomalacia, okay, we have biotot spots, white dry eye, okay, and then follicular hepatokeratitis we have. In this there will be phyroderma, toad, okay, and also in this there can be vaccines also, like OPV vaccine, rota vaccine, vitamin A. So all these things we can see. Then comes about vitamin C. Vitamin C, this is the most important because maximum of the questions comes from here. Vitamin C, this is ascorbic acid. Okay, C means, vitamin C means ascorbic acid, C. Then C means citrus, right? It is also antioxidant. C means collagen. C means scurvy. SC will come in all. Okay, in this the PT will be normal. Okay, as I already told this also, PT is the most important because in some of the cases it is increased, in some of the cases it is decreased. In vitamin K it was increased, in C it will be normal, and it helps in absorption of iron. This question has also come, vitamin C. Okay, in this there will be microcytic hypochromic anemia. This question has also come. In this there will be defect of collagen, bleeding manifestation. Okay, and also in this there can be like iron deficiency anemia, thalassemia, anemia coagulatis, cetal. So like this, all these things will be there. And vitamin C there will be T, tannic T will be there, and phytate absorption inhibition. All these things will be there. Then comes vitamin B. B1 is thiamine. Okay, in thiamine what do we have? We have transketolate enzyme, and the coenzyme which we have here is TPP, thiamine pyrophosphatase. Okay, and here we have is pyruvate dehydrogenase, and then we have coenzymes alpha ketoglutarate, which I dehydrogenase, which I told, that leads to what? This leads to lactic acidosis. Okay, and in this we have branch chain keto acid, pyruvate, we have dehydrogenase complex like TPP, NAD. So all these things are there. In lipoic acid we have vitamin B, and in this we have this increased uptake, which is spinach, broccoli, potato, yeast, and this will prolong the deficiency. And so these things, spinach, they all prolong deficiency. And then we have in B1, they can ask which is the least likely to cause, that will be beriberi in the CNS. And also in the B1 there will be polished rice. Okay, then you all know about Wernicke encephalopathy, Goa, global confusion, ophthalmologia, ataxia, Korsakoff, you have scap, confabulation, amnesia, polyneuropathy. Then comes vitamin B2, we have riboflavin. B means you can understand. Then comes flower, that is flavin. Then in this we have Warburg yellow, enzyme of respiration. Remember one thing, in vitamin B1 what do we have is transketolase. Here we have glutathione reductase. So do not confuse here. And in this what do we have here is we have coenzyme, then it's FAD. This question has also come. In dehydrogenase enzyme what will be there? So FAD and FMN. In vitamin B2 FAD and FMN is there. Then comes vitamin B2 whenever it decreases. So what all things we can see? In lips we can see chelosis, angle of mouth, angular stomatitis. Then comes tongue we have glossitis. Okay, and it increases cornea vascularization. Okay, then comes vitamin C. C means nicotinic acid. Okay, TNIC means tryptophan, and vitamin B3 means it's 3, right? Capital C. Then it will be niacin. Okay, then vitamin B3 what will be there? NAD coenzyme will be there, dehydrogenase complex. So in B1 what was there? In B1 it was polished rice, but in B3 it will be maize, polished rice and maize. Then comes tryptophan. Tryptophan what, whenever it decreases due to excess of the leucine. Okay, vitamin B3, and then that lead to pellagra. Okay, and also about I am telling you just little bit more. In this there will be Hartnum disease and carcinoid syndrome. Okay, these all things can be there. Then comes Castle's necklace. In this there will be dermatitis, diarrhea, dementia, death. All these things can be there. And also in this there can be macular edema, macular cyst, because these are also related to vitamin B3. Then also it can cause hyperglycemia and diabetics, right? You all know casus necklace, pulses we have cysteine, methionine, cereals we have lysine. Okay. Then we have vitamin B5 means 5 means 5, 5 means pantothenic acid. Then comes coenzyme is coenzyme A. Okay, this is also called as burning foot syndrome. This is very important. Okay, then comes 6. Okay, do not confuse with vitamin B3 and B6, because B3 is tryptophan, nicotinic acid and niacin. Here it will be pyridoxine, 6 SIX and pyridoxine XI. You can add both of them, and also XI is there. So from X only you have xanthuranic test, which is also called as tryptophan load test. This is the one which is not required in TCA cycle, and in this we have coenzyme. Okay, pyridoxal 5-phosphate. Then it's very important in TB, isoniazid. Okay, and when this decreases, there will be sideroblastic anemia and seizures. Okay, and also I told you about one more anemia. Okay, in which I told about hemolytic anemia. So whenever you have hemolytic anemia, then it will be vitamin E. Okay, and whenever there is sideroblastic anemia, then it will be vitamin 6. Also that will lead to seizures, sensory neuropathy. Okay, this question has also come. And the coenzyme which was PLP, which is pyridoxal phosphate, this question has also come. Okay, then coenzyme which we have is this PLP, which I told. And yeah, this is done. And let's talk about vitamin B7. In B7 we have biotin, because B is there, so biotin. And B, after B what comes is C, so carboxylase. Okay, so when it comes to carboxylase, so the reaction which will be there here will be also carboxylation reaction, right? And also we have here is like gluconeogenesis, fatty acid, propinic acid pathway. We have question is about gluconeogenesis. So neogenesis, there will be pyruvate carboxylase. Okay, and acid, fatty acid, then it acetyl, propionate, propional coenzyme carboxylase. Okay, then comes egg. Whenever comes egg, think of vitamin B7. When it comes of avidin, think of vitamin B7. Okay, avidin plus biotin, this will decrease absorption of biotin. Okay, then comes 3 things are there: iron, B12 and folic acid. Okay, iron, B12, folic acid. And also we have 3 things in sequence: duodenum, jejunum and ileum. But what we do here is duodenum, ileum and jejunum. So iron, B12, folic acid, duodenum, ileum and jejunum. Okay, so this is how you have to remember. And you can also remember this at the end you have fufa and jija, right? Fufa is folic acid, jija is jejunum. Then comes folic acid. Okay, about folic acid, which I told you about jejunum. So let's see. In this, first of all, there will be vitamin B9 deficiency. Okay, here it will be megaloblastic anemia. And can someone tell about vitamin B6? In B6 it was sideroblastic anemia. And about vitamin E, E was hemolytic, B6 is sideroblastic. Okay, and B9 will be megaloblastic. Okay, hemolytic and then megaloblastic. Okay, so here it will be macrocytic anemia. Okay, in this there will we give 400. See, it's prevention, right? Just remember, whenever there is a prevention, it will be 400 microgram. Okay, in order to prevent neurotoxic defect, which is in vitamin 9. But whenever it's pregnancy and when they ask about treatment, then it will be 500 milligram. Okay, so please do not forget it. And in this there will be test FIGLU, histidine. Okay, FIGLU test is there. And in this there will be 3 things are there. For example, we have food, then it comes to folic acid, then folate hydrogenase 2, dihydrofolate, tetrahydrofolate. Then comes 3 things: either DNA, it will form either mecobalamin, or either it will be homocysteine. So question has come that these all are what? So these all are 1 carbon atom. Okay, now comes about B12 and B9. So we need to know the difference between both of them. See, in B12 there is folate trap. Okay, and whenever B12 decreases, see there are four factors: either megaloblastic anemia, neuropathy, homocysteine, and methylmalonyl. Okay, so the differentiating feature which we need to know about are the last two as important: homocysteine and methylmalonyl coenzyme A. Because megaloblastic anemia is same in both of them, vitamin B12 and vitamin B9. So this is same. Neuropathy will be there in vitamin B12. This is the first point. Second is about homocysteine. Homocysteine will be equal in both of them. Okay, this will also be same, but methylmalonyl coenzyme A, this is the one which will be increased in B12, decreased in B9. Not decreased, it's normal, right? So there are two factors which are increased in B12. First is neuropathy, second is methylmalonyl coenzyme A. And in B9, there are two things. See, megaloblastic anemia is also positive, and also homocysteine is also increasing. Two things are differentiating feature. Neuropathy will not be there. Methylmalonyl coenzyme A will be normal. Okay. In B9, we have Figlu test, histidine load test. Okay. In B12, we have it's come from animal product. Okay, and also in this we have a test called Schilling test. Okay, and also in this there will be methionine synthase, that will be methylmalonyl coenzyme A absorption. Okay, so these are all the things about vitamins which I have tried to cover in very conceptual way. Okay, now let's see about nucleic acid. Okay, in nucleic acid, what do we have here is we have double strand, helical, polymer, pairing, antiparallel. What you need to know: nucleoside and tide. Side means S. S means 2. So there will be two things: sugar and nitrogenous base. Whenever tide means three things will be there: sugar, nitrogenous base, and phosphate. Now you need to know what is this nitrogenous base. Which sugar I am adding? One nitrogenous base. So what is this nitrogenous base? Divided into two: purine, pyrimidine. Purine is adenosine, okay, guanine, hypoxanthine, xanthine. These are purine. Pyrimidine is CUT. Okay, C is cytosol, uracil, thymine. Okay. Now comes nucleoside and nucleotide. Okay, what is side and tide? Which I told about, one is 2, one is 3. So in tide, you know, both and 3 we have added with phosphate. So whenever we have adenine and guanine, okay, so what are they? They are purine. Adenine and guanine are purine. Okay, and cytosol, uracil, thymine, these are pyrimidine. Okay. Now first of all, in purine there will be end product. If they ask about end product, it will be uric acid. Okay. And in pyrimidine, if they ask, there is the most common. This is orotic aciduria. Remember this word. Okay. In adenosine there will be sine, adenosine, guanosine, and that is gene, right? Then we go ahead and we see a model called as Watson and Crick model. Okay. In this, what is there? We have phosphate, sugar, nitrogenous base. Okay, and we attach with a bond which is hydrogen bond. Okay, and then we have again one more bond in which we have phosphate, sugar, but in right now when it was A means adenosine. Adenosine will bind with T, which is thymidine, and whenever it's G is guanine. Guanine will have a bond with whom? Cytosine. GC, ATGC, right? Okay. And also one more thing which I am telling you right now only, that in that there will be cycle like mitochondria and cytosol will be there, right? So what will happen is CO2 and NH3, they both will combine in mitochondria, and there will be carbonyl phosphate synthase that will lead to carbonyl phosphate that will lead to urea cycle. So they will ask about whether it's type 1 or type 2, the one which is converting. So if carbonyl phosphate synthase 1 is there, then that will be of mitochondria and that will lead to urea cycle. But if we have CO2 plus glutamine and that is converted into pyrimidine synthesis, then that means there will be type 2. So in urea it was type 1, and here in pyrimidine it will be type 2. Okay, and let's see about the rest. We have Chargaff rule. Okay, what is there? It's nothing, just you all know ATGC. Okay, A plus G equal to C plus T. Okay, and then we have types of DNA. Watson crick we have left hand helix we have. Okay, then we have right hand helix. Okay, so all these things are there. About just little some points I am telling you regardless of this topic. See, we have CRISPR gene. CRISPR gene means it is a gene editing. Okay, just edit the gene. These are the direct questions which have already come. CRISPR gene means gene editing, and whenever you have APO B48, that means it is RNA editing. CRISPR means gene editing, APO means RNA editing, and whenever you have cellular DNA in mitochondria, that is 1%, and whenever you have cytoplasmic, sorry, then it will be S adenosine methionine universal methyl group. Okay. And if they ask you about subunit aggregation, then you have hemoglobin, collagen, and Ig. Okay. And whenever you have testosterone, so source of testosterone is cholesterol, and what does it produce? It produces Leydig cells, right? Then we have some phases. One phase is we have linear DNA. If they ask about regulatory gene, tell repressor. If they ask about point mutation, say sickle cell. If they ask about methotrexate, then it will lead to inhibition of dihydrofolate reductase. Okay. Now let's continue towards the DNA one. Then we have denaturation and renaturation. Whenever your denaturation means bond is breaking, temperature and pH is increasing. Renaturation means there will be bond is forming, temperature, pH will decrease. Okay. Then comes nucleosome. In nucleosome we have many things: histone, protein, DNA. Okay. And then we have is octamer. Okay. In octamer we have 8 monomers. So like we have H2A, H2B, H3H4. I know right now it must be looking simple, but it may confuse you in exam. So the one which is not here is H1. Just remember one thing. It will be octamer 2H2A, 2H2B, 2H3, 2H4. In equal to it will be 8, but not will be there. No H1 will be there. Okay. Then comes replication. Okay. So whenever DNA is replicating, so in DNA polymers we have 3 types: 1, 2, and 3. Okay. It's usually there in prokaryote. Question has come. It is single-stranded DNA. Question has come. Okay. It is there in mitochondria. Question has come. It is there in S phase. Question has come. Okay. And in this there will be bacteria and blue-green algae. Okay. And all others are eukaryotes. Then comes helicase. Okay. Then like we have in DNA application we have the diagram. In this first we have leading phase, lagging phase. Okay. 5:3 is leading, and okay. Then we have a lagging phase. Then we have fragments called Okazaki fragments, and also later on it leads to recoil, supercoil. Okay. Then we have helicase. So what is helicase? It is the one which uncoils it. Okay. Then comes separation of the strand. So who separates the strand? Prevents separation from annealing. Okay. If we need to separate the strand, then it will be AP protein. AP protein separates the strand. Separate means PA is there, na? So PA means P is protein, A is A, so DNA A protein. Helicase is uncoiling. Stabilizing means SSBP. Okay. Means single stranded binding protein which stabilizes it. Then comes supercoil. Who is supercoiling it? It is topoisomerase, right? Then come new strand. So we have 2 strands, continuous and discontinuous. Continuous is what? Continuous is the one which is leading strand. Okay. It is formed by DNA P3. Okay. So whenever you have DNA P3, that is a leading strand, 5 to 3. Okay. What is discontinuous? Okay. Leading is continuous, lagging is discontinuous. This is also the same DNA P3, and this will also be same DNA 5/2 3/. But in the discontinuous one, there will be some fragments which I told is Okazaki fragment, and they join which ligase? DNA ligase 1. Okay. Then we have RNA primer which is filled by DNA protein 1. Okay. DP1. Okay. So we have Okazaki fragments which join and form DNA ligase 1, and whenever it's RNA primer, okay, it is also DNA protein one, but not ligase, right? Then comes DNA repair. Which repair? See, if they ask about repair, then it's 2, DNA P2. Okay, whenever it's RNA, DNA P1. Whenever it's discontinuous, DNA ligase 1. Okay, then comes eukaryotic. In eukaryotic, what do we have here is alpha, beta, gamma, delta, epsilon. Okay, in alpha, what do we have? Primase, prime number hota hai alpha. Then comes beta. Beta is always repair. Okay, beta we have in our Indian society, we have beta. So what is its work is to repair family or anything. So it will repair DNA also. Then comes gamma. Gamma is girls. Okay, so girls are like, they usually they are almighty. They talk to almighty a lot. So mighty. Okay, mighty means mitochondria. Okay, they are more like devotional, so they talk to mighty, almighty, right? Mighty means mitochondria. Then comes delta. Delta means donkey, and E means epsilon. Epsilon means elephant. So whenever you have donkey and elephant, if they fight, who will lead? Who will win? Obviously, elephant will win. So epsilon is leading, and delta is donkey. Donkey will be lagging. Okay. Then comes transcription. Transcription, I already told, DNA to mRNA. Okay, it is DNA dependent. In this, there will be histone acetylation, DNA methylation. A question came in which they told about transcription. It was histone acetylation, DNA methylation. So there will be DNA, and 3 to 5 will be there in DNA. And in this, there will be a question has come out, transcript. What is primary transcript? Primary transcript is the one which convert 5/ to 3/. Okay, so usually it was 3 to 5, which is DNA template, and whenever it's RNA it's 5 to 3, and this is primary transcript, right? Okay, then there is a gene, we all know that it is dystrophin gene, and in this it is not AT, it will be AU. Okay, then comes prokaryote, eukaryote. See, you just later think of prokaryote and count the letters. It will be 10 letters. So 10 sequence will be there. 10 means Tata box. Okay, and that is called as Pribnose box. Okay, it was prokaryote, and eukaryote means E means human. Okay, letter number is 5. 5 means 25 sequence. Okay, human is Hogness box. Okay, then comes RNA polymerase. In RNA polymerase, we have 3 things. Okay, remote. We remember it by remote. Okay, REMOTE. So question comes about usually all except. So R is RP1, M is RP2, which is really M, 3 means T. So in RP1 we will have rRNA, which is the most abundant, and RP2 we have M is mRNA, and RP3 will be tRNA, tiny transfer of amino acid. Okay, then in tRNA, what do we have? T is the one which is tiny and which is transfer of amino acid. Okay, then in this there will be clover leaf shape, there will be acceptor arm and anticodon. Okay, and now comes our protein and pyrimidine. Sorry, purine and pyrimidine. In purine, in RNA it will be 2. Pyrimidine also there will be 2. So they will ask about AUG. So we have 2 and 2, 4. So we do 4 cube root, that will be 64. Okay, and in total they will ask about the codons, how many codons have, then it will be 64 codons. And if they ask about genes, codon we have is 64, genes we have 20 to 25k genes. Okay, then we have single acid which can be coded by more than one codon. See, they can ask about the definition. What is degeneration? If a single acid which is coded by more than one codon, then it's degeneration. Okay, when a code there is a code, okay, which is only done by a single amino acid. Okay, if single amino acid is coded by more than one codon, degeneration. If few codon are coded by only one amino acid, then it is specific, right? So whenever it's specific, that will be unambiguous. Okay, in this there will be AUG and CCU. Okay, and it's universal and also it's commonless. So this has also come in all except question. Okay, now let's say about AUG. AUG is methionine, which is we also call it start codon. Then we talked about 21 and 22. 21 is Selena Gomez. Okay, UGA, and then comes 22 pyro. means fire, which was us, it was pyrolysin. Okay, there it was UGA, here it will be UAG. Okay, then comes translation. Translation is what? mRNA, tRNA. Okay, then comes AUG, initiating codon. Okay, AUG is initiating codon, which we know, which is methionine also we say. Then we need to know about stop codons. Okay, in stop codon we remember it as CAMP. Okay, Ochare, Embal and Opal. Ochare, Embal and Opal. So means we have any codons, but remember it as Ullu Aya, Ullu Agaya, Ullu Gaya. Ullu Aya, Ullu Agaya, Ullu Gaya. Okay. Then comes ribosome. We have prokaryote, eukaryote. Prokaryote is 70, eukaryote is 80. Then we have RNA. It's linear. AGCU, phosphate ribose we have, and here we have is fish. This is very important. Okay, its intermediate is histidine, and in this we need to know about what is fish. It is fluorescent in situ hybridization. Okay, this in this the choline is avoided. Okay, and in this what is this? This is abnormal locus and chromosome structure abnormality. Okay, in this there is PCR, polymerase chain reaction. Okay, and amplification will be DNA. Okay, so in this there are three things. First of all, what we do is denature DNA, then it will anneal primal, and then extend the DNA. Okay, then a question comes about RNA virus. This is very important. RT-PCR, monkeypox will be there in it. Okay, then we have RNA virus that will lead to reverse transcription, and that will lead to DNA which will be amplified. Okay, so in RT-PCR, RNA to DNA which will be amplified. Usually coronavirus, monkeypox, all these things are there. Then we have three blots. Okay, we have western blot, southern blot, northern blot. Then in west we eat pizza, so then it will be protein. South we eat dosa, then it will be DNA. In north we eat roti, that is RNA. Okay, in real time PCR amplification plus quantate. So this is also question, xeroderma pigmentosa. What is this? Nucleotide excision repair. Okay, and it is separated by electrophoresis, southern blot. Okay, let's start amino acid now. In this we have is glycine, which is simple amino acid. Most common amino acid is collagen. Okay, then we have proteins, band fold proteins. Then we have animated transmitters. We have conjugation, heme synthesis. In purine ring we have aspartate, glycine, glutamate. Okay, in aspartate we have adenine, glycine, glutamate we have guanine. Then we have glutathione. Okay, gluta means glutamic acid. Okay, one means ring. Okay, then it will be glycine. Then we have creatinine. In creatinine, what do we have here is glycine, arginine, methionine. Okay, so in glycine, what do we have here is primary hyperoxaluria in glycine. In creatinine, we have cricket. Okay, from cricket, what do we play? Whenever we are there in cricket, we do argument and we play the game. So cricket is creatinine. Okay, game GAME. GA is glycine, ME is methionine, argument is arginine. Okay, which is primary hyperoxaluria. Then comes heme synthesis. Heme means what? Glycine plus succinyl coenzyme A. That will lead to ALA synthase. Okay, alanine synthesis, which is a rate limiting enzyme of heme synthesis. Okay, and also this decreases vitamin B6, which lead to sideroblastic anemia. Okay, ALA means amino levulinic acid. Okay, then what will we do it? It will reduce H2O with ALA dehydratase, right? Then it will lead to porphobilinogen. That will lead to three things: uroporphobilinogen, corpopyrofrenogen, protopyrofrenogen. These three things are there. And then protoporphyrin, that will lead to ferrochelatase and heme. So we have lead. Okay, whenever lead will inhibit, it will inhibit what? ALA dehydratase. Okay, and it will inhibit, second thing is ferrochelatase. Okay, this will also help in ferrous. So lead and ferrous, two things will be involved in it. Okay, heme synthesis. Then comes heme plus globin is 4 pyrrole ring, beta alpha alpha beta. Okay, F2 plus. Then hemoglobin will denature bilirubin, right? Then we have transport of neutral acid. How it is transported? We have intestine. Okay, in intestine we have leucine, so we have Meester cycle. In Meester cycle, what do we have? Meester cycle, remember word G. G is glutathione, gamma glutamyl cycle, GGT, liver and kidney. Okay, so in Meester cycle we have 3: glutathione, gamma glutamyl cycle. Okay, and then comes in neutral amino acid, which was in intestine. Then come 3 things: GAL, GAL. G is glucose, A is alanine. Okay, and the cycle here is Hill's cycle, CA Hill's cycle. Okay, so this was all about neutral amino acid, GAL, glucose, alanine, CA Hills. Then we come about urea cycle. Urea cycle will be there in both cytosol plus mitochondria, which will be there in liver. Okay, then we have here is it is also called as Krebs-Henselt cycle and ornithine cycle. Okay, so what we need to know here is, first of all, the rate limiting enzyme, which is CPS1. CPS is carbomy phosphate synthase 1. Okay, CPS1. And in this we have a mnemonic called as occasions are for argument. There it was game, cricket. Okay, we had argument, then we played a game. Here it's like occasions are for argument. We have ornithine cycle. Okay, as an occasion we have O is ornithine. Then we have C. Second C is carbamyl phosphatase, which increases the glutamate level. Okay, then we have second C, occasions. Second C we have citrulline, ornithine, carbamyl phosphate, citrulline we have. Then we have A is aspartate. Okay, AS we have now occasions. A is aspartate. Okay, then occasions are done. Then ARE. Okay, then AR is arginosuccinate. Okay, then after arginosuccinate we go down and we see what in arginosuccinate we have AR is there, so use the AR 2 times, arginosuccinate and arginine. Okay, then comes 4. Occasions are 4 is fumarate. Okay, and then then comes what? Then we have AR is also done, arginine, and then comes argument. So GU, U is urea, which is an excretory product. Okay, and then arginine, from arginine comes urea and ornithine, and again it becomes ornithine, right? So in carbamyl phosphatase, glutamate level was increased. Okay, in arginosuccinate, citrullinemia will be there. Okay, in fumarate there will be urea and Krebs cycle both, and urea is also there as an excretory product. Okay, and also they can ask you something, which one is the common precursor in arginine. Okay, so common precursor will be urea, creatinine, nitric oxide, UCN. Okay, and also one more point I would like to add here is urea and Krebs cycle, they are linked at what? They are linked at fumarate, which I told, and then comes ornithine. So what's the, in ornithine what will be there? This will increase atrophy of cornea and retina. Okay, then comes NH4, NH2 plus CO plus NH2. These 3 things were there. Okay, so in NH2 and all these things, this will lead to aspartate. CO means CO2, NH2 means NH4. Then comes ammonia. What does ammonia do? From ammonia this will go to brain, from brain to liver, from liver to glutamine. Okay, then comes tryptophan. What is tryptophan? PLP. In this there will be vitamin B3, niacin. Okay, in this there first of all there will be 60 mg. Okay, that will lead to decreased absorption of intestine. Okay, and in tryptophan, okay, what will be there? Just remember, tryptophan is H is there na, HAN. HAN means remember H1, A2, TR, that is 3. So like this, if you see that will lead to heart-knob disease, that will decrease niacin, and that will lead to pellagra. Okay. Now phenylalanine. In phenylalanine, what is there? Tyrosine and melanin. Okay. In tyrosine and melanin, what will be there? In this, there will be phenylalanine hydroxylase will be there. Okay. Then phenyl ketonuria will be formed, where in phenylalanine, okay, that will lead to which test? Glutaric test. Okay. And also in this, there will be ferric chloride test. Phenylalanine, remember this because question has already come. They will ask about where will be PKU test, phenylketonuria, you need to have phenylalanine test. Okay, glutaric test, also you need to have ferric chloride test, also it will be same, phenylalanine. Symptoms, yeah, symptoms will be mental retardation and musty or mousy smell. Okay, so this will be phenylalanine. And now comes tyrosine metabolism. What will be there? In tyrosine, there will be homogentisate oxidase. Okay, then comes fumaryl acetoacetate. All these things will be there, and then that will lead to L-captonuria. Okay, that will increase homogentisate, that is black coke urine. Okay, that will lead to cartilage and pennine sclera, and also that will lead to ochronosis, which is L-captonuria. Usually in L-captonuria, they will ask a 35-year male, back pain, pigmentation, IQ is normal, what it could be? L-captonuria, and there will be nitiso. Okay. So this was all about L-captonuria. Now let's see what is albinism. In this, first there will be tyrosine. Tyrosine will convert in tyrosinase to melanin, and then in this albinism there will be depigmented skin, photophobia, hair white, nystagmus. There will be Shadia-Higashi syndrome, which will be in albinism and neutrophils. Okay, and then comes about tyrosine. Okay, further about tyrosine. First of all, we need to know the rate limiting exam. This question has already come in phenylalanine. That is catecholamine. Sorry, rate limiting enzyme of catecholamine also, and this will be tyrosine hydroxylase. Okay, tyrosine hydroxylase. Then there will be dopa. Then will be dopamine. Then there will be norepinephrine, and then epinephrine. Okay, so what are these? These are catecholamines. What is catecholamine? Dopamine, epinephrine, non-epinephrine. Okay, so this will be catecholamine. This as question has also come. So catecholamine rate limiting reaction can also be the same, tyrosine hydroxylase. Okay, then we have tyrosine. In this there will be T3 and T4. In tyrosine there will be no melatonin as a precursor. Okay, T3 and T4, that will lead to thyroxine. Okay, so T4 is 90%, T3 is 10%. Then come thyroid. Thyroid have what? Follicular and parafollicular cells. Parafollicular cells. Okay, follicular cells means just take out one more C of follicular seed, and then that will lead to then we have parafollicular cell, that will lead to calcitonin, and in this there will be, just a moment, and then whenever we have follicular cell, that will be T3 and T4. Parafollicular means calcitonin, right? Yeah, so whenever we have T3 and T4, they are formed with the help of sodium iodide symporter, right? Now there is a table. In this table, we need to see like which is converted into what. So we have like 3 products, 1, 2, and 3. First will be fumaril, second will be TT, third will be PPP. Like this, things are there. So in first one we have like albinism, phenylalanine hydroxylase. Second we have tyrosine transaminase. Okay, and in third one we have PPP is phenylpyruvic acid oxidase. Okay, so first one was fumaril, right? So that will in this there will be hydroxylase type 1. In TT, which I told, TT will be oculocutaneous. Okay, and in third one will be least severe neonatal tyrosinemia. About fumaril, I would say that in this there will be also kidney problem will be there, severe kidney problem will be there, and hepatorenal. Okay, then comes first fumaril hydroxylase, which I told about first. In this there will be branch chain amino acid, question has already come. Okay, and in this there will be valine means valine leucine isoleucine. Okay, in type 1 there will be decarboxylation. Okay, branch chain amino acid V Wali and decarboxylation, three things will be there, and that will lead to branch chain keto acid dehydrogenase. Okay, this will be there, and as a product it will be maple syrup disease, burnt sugar syndrome. Okay, and in this there will be MSUD, we will study later on, and that will be dihydrolipoyl transcyclase. Okay, so about fumarate hydrolase I told, branch change amino acid. Okay, Wali VALI. Okay, branch changed keto acid, then product is maple syrup or burnt. Then comes propionic acid pathway. In propionic acid we have source, we in this we can remember it by mnemonic. Word meal, word means V means valine, O means orchin fatty acid, M methionine, I isoleucine, T threonine, through. Okay, in propionyl, what do we have here is we have carboxylase. Okay, and then we have methylmalonyl mutase, succinyl coenzyme A. Okay, then come a question from here. What it is asked is cystinuria. Excrete amino acid in urine. Okay, first of all, it is not cysteine, not E, it's U. Okay, so cystinuria. So in this, what will be there? Four things will be there: cysteine, ornithine, lysine, arginine. There will be no E, only I. Cysteine, ornithine. Okay, so that we can remember it as cola. COLA. Cystinuria means cola. Cysteine, ornithine, lysine, arginine. Okay. Then we have semi-essential amino acid, which I told, histidine, arginine. So a question has come about histidine means you decarboxylate, it will lead to histamine. But when you have arginine, in arginine that will lead to nitric oxide. Okay, that will be VD. So arginine converts with NADPH to nitric oxide. This question has already come in semi-essential amino acid. Then comes salvage pathway. Okay, in this you have purine metabolism, PAGX. Okay, and in this there will be like we have adenosine, guanine, hypoxanthine. Okay, all these things are there. So adenosine will convert to AMP. Okay, guanine will convert to GMP, HGPRTase. Then come hypoxanthine, HGPRTase, that will convert to IMP. Okay, so XO is xanthine. Okay, xanthine oxidase will convert and will convert to like guanine to GMP, hypoxanthine to IMP. So like this, this will be converting. Okay, now comes a very important thing from this question has already come, and that is hypoxanthine guanine phosphoribosyl transferase. So in this we have Lesch-Nyhan syndrome. Okay, that is due to HGPRT. Okay, and in this there will be increase in uric acid, which will lead to gout. Also in this there will be neurological symptoms present. There will be self-mutilation, that means child will be there biting or chewing of lips or nails. Okay, and drug of choice will be gout. And in this the chronic gout, if it is chronic gout, then we give xanthine oxidase. Okay, NSAID we give, then xanthine oxidase. Okay, and whenever we have in this adenosine, adenosine. Okay, adenosine deaminase, then iosine. So we need to focus about adenosine deaminase, AD. DD are there in both, adenosine ka D, deaminase ka bhi D. Take both of the D and just write SCI in front and SCID. It will form a SCID. Okay, adenosine deaminase. So severe combined immunodeficiency. Okay, in this there will be T plus B cell decrease. Okay, and whenever it's a defect, then it will be non-homologous and joining repair. So see, whenever you have a defect in SCID, then that will be non-homologous and joining repair. But whenever you have a problem in xeroderma pigmentosa, in that there will be nucleotide excretion repair. But here, non-homologous and joining repair. So this was all about amino acid, and we talk about more about lipid.